Before any scientist carries out a microbiology test, they must prepare themselves, their equipment, and their laboratory correctly. Practical activities in T-level science provide an excellent opportunity to teach students not only the scientific technique itself, but also how to prepare for practical work safely and professionally.
By the end of this lesson, you will be able to explain the purpose of standard operating procedures, describe how gram staining differentiates bacteria, identify contamination risks and how to prevent them, and explain the importance of calibration, quality assurance, and quality control. You'll also understand why confidentiality and data protection are essential in scientific workplaces.
To be successful in this lesson, you should be able to define key scientific terms such as SOP, quality assurance, quality control, contamination, calibration, and confidentiality. You should also be able to explain why laboratories follow standardized procedures, identify gram-positive and gram-negative bacteria, recognize workplace responsibilities in scientific organizations, and apply your knowledge to a real microbiology workplace scenario.
For today's lesson, you will need a range of microbiology resources, including the gram stain SOP, microscope, bacterial culture images, slides and cover slips, staining reagents, immersion oil, calibration equipment, and appropriate PPE. These are the same types of resources used in professional laboratories to ensure scientific work is carried out safely, accurately, and consistently.
Gram staining is one of the most common techniques used in microbiology laboratories, including NHS pathology, pharmaceutical, food testing, and water testing laboratories. It is a differential staining technique used to distinguish between different groups of microorganisms. By identifying differences in bacterial cell walls, scientists can classify bacteria quickly and support diagnosis, quality control, and scientific investigations.
Imagine a patient arrives at hospital with a serious infection. Scientists need to identify the type of bacteria as quickly as possible so the correct treatment can begin. Gram staining helps microbiologists distinguish between different groups of bacteria within minutes, making it one of the most important diagnostic techniques used in laboratories around the world.
Before any practical activity begins, scientists must follow a standard operating procedure, or SOP. And SOP provides clear instructions to ensure work is carried out safely, accurately, and consistently. By following the same procedure every time, laboratories maintain high standards, comply with regulations, and produce reliable results that can be trusted.
As you review the SOP, look carefully for the SOP number, version number, review date, and signature section. These details help ensure that staff are following the most up-to-date procedure. Laboratories require the latest SOP version to maintain consistency, improve safety, meet legal requirements, and ensure that results are accurate and reliable.
Although bacteria may look similar under a microscope, they have important differences in their cell wall structure that cannot be identified by appearance alone. Gram staining is a differential staining technique that separates bacteria into two groups. Gram-positive bacteria have a thick peptidoglycan cell wall and appear purple, while gram-negative bacteria have a thin peptidoglycan wall and appear pink or red after staining. This helps scientists identify bacteria quickly and accurately.
During the demonstration, follow each stage of the gram staining procedure and record the steps in your flow diagram. Notice how crystal violet, iodine, ethanol, and safranin each have a specific role in differentiating bacteria. Following the correct sequence and timing is essential to produce accurate, reliable results and avoid misidentifying bacterial samples.
Ethanol is often called the critical step in gram staining because it determines whether bacteria are classified as gram-positive or gram-negative. If too much ethanol is applied, gram-positive bacteria may lose the crystal violet stain and appear gram-negative. If too little is used, gram-negative bacteria may remain purple. This demonstrates why scientists must follow SOPs carefully and use precise timing to ensure accurate and reliable results. At Station 1, investigate the light microscope and identify its main parts. Consider its purpose in microbiology and discuss why immersion oil is used when viewing bacteria at high magnification. Focus on how the microscope helps scientists produce clear and accurate observations.
At Station 2, explore the eyepiece graticule and stage micrometer. Complete the worksheet and discuss why scientists calibrate measurements before collecting data. Consider how calibration improves accuracy, reliability, and quality assurance in laboratory work.
At Station 3, investigate the role of the incinerator and autoclave in microbiology laboratories. Discuss why equipment must be sterilized before use and consider the consequences of poor sterilization, including contamination, inaccurate results, and potential risks to health and safety.
At Station 4, examine the 4 reagents used in the gram staining process and match each one to its function. Consider how crystal violet, iodine, ethanol, and safranin work together to differentiate between gram-positive and gram-negative bacteria. Understanding the purpose of each reagent helps ensure the procedure is carried out accurately and consistently.
In this activity, you will act as laboratory auditors. Review the scenario carefully and identify any breaches of laboratory procedures. Consider the impact of not wearing PPE, using expired reagents, failing to label slides, and leaving equipment unclean. Discuss how these issues could affect health and safety, result quality, and compliance with laboratory standards and SOPs.
Working in your groups, identify the issues within the laboratory scenario and discuss their consequences. Consider whether each issue represents an SOP breach, a health and safety concern, a quality concern, or an audit finding. As you discuss, link your answers to SOP compliance, quality assurance, professional standards, and audit readiness within a scientific workplace.
Scientists must communicate their observations clearly and accurately. Using the image provided, produce a scientific drawing of either gram-positive Kochi or gram-negative bacilli. Remember to include a clear title, accurate labels, magnification, and neat lines without shading. If you finish early, use the graticule calibration data to estimate the size of the bacterial cells, just as microbiologists would in a professional laboratory.
Before you leave, complete your exit ticket to check your understanding of today's lesson. Think back to the gram staining procedure, the role of each reagent, contamination control measures, and the importance of SOPs in laboratory practice. Your answers will help identify what you've learned and any areas that may need further review.
As we finish today's lesson, remember that successful microbiology relies on more than just scientific knowledge. Differential staining is essential for identifying bacteria, but accurate results also depend on following SOPs, using PPE correctly, calibrating equipment, preventing contamination, and maintaining high-quality standards. Professional laboratory behavior is just as important as scientific technique in ensuring safe, reliable, and trustworthy results.
Thank you for watching. We hope this lesson has provided useful ideas for delivering practical skills in T-level science. Follow the channel for support with practical activities, lesson planning, ESP guidance, exam preparation, and industry placements. Don't forget to subscribe and turn on notifications so you never miss a new T-level science video.